Hello and welcome to Biotech 101 Short Video Explainers by Biotech Blueprint. This week we'll talk about gene editing and CRISPR and how this technology just saved a baby's life. What is gene editing? Our bodies are made up of about 35 trillion cells, which are the smallest living units of life, and they do all different things in the body. Cells have tiny organs, they're organ-like structures inside them that we call organelles. And one of these organelles is a very important one called a nucleus, which contains the DNA. The DNA being an instruction manual for life, basically. The DNA is packed into 46 chromosomes that carry genes, and genes are these tiny sequences that act as recipes on how to make proteins. These proteins then do nearly everything in your body that keep us alive and healthy. Sometimes, though, these recipes can have a typo, and the typo, which is a tiny error in DNA, can cause devastating diseases. Gene editing is the science of fixing those typos, and CRISPR is a tool that scientists use to do it. In 2012, scientists Jennifer Doudna and Emmanuel Charpentier transformed gene editing. They discovered how to repurpose a system called CRISPR, which actually is a natural immune system of bacteria. Bacteria can also be attacked by viruses. They're called bacteriophages. And they actually have this CRISPR thing where they cut up the viral DNA and protect themselves from this assault. And so they discovered how to repurpose these molecular scissors into a tool for editing DNA. This system called CRISPR is made of a guide RNA, which actually scans the genome and finds the specific gene or the specific spot in the DNA. There's also the enzyme, that important part, called Cas9. In this case, we for now use mostly Cas9, and it's basically a nuclease that acts as the molecular scissors that makes the cut in the DNA. And then scientists can either delete or disable or rewrite a gene, basically put in a gene of interest instead of the faulty gene. It's very precise, it's powerful, and in 2020, Doudna and Charpentier won the Nobel Prize in Chemistry for this discovery. Last week, on May 15th, 2025, it was announced that scientists used CRISPR to help save the life of a baby boy named KJ. KJ was born with OTC deficiency, which is a rare genetic disorder that causes toxic ammonia to build up in the body, and it can often be deadly. In just 6 months, researchers designed a custom gene editor. They tested it and delivered the treatment to KJ, which was targeted to his exact mutation. It wasn't a complete cure, but it worked. His symptoms improved and his ammonia levels dropped significantly. So this is called personalized gene editing. It can be delivered in real time, and it really opens doors to treating ultra-rare diseases with precision. In 2023, the CRISPR-based therapy called Casgevy, developed by CRISPR Therapeutics, was approved to treat sickle cell disease. And doctors take patient cells, stem cells, they're called hematopoietic stem cells, that then go on and make red blood cells, because sickle cell disease is ultimately a red blood cell disorder. And they edit these stem cells outside of the body and then infuse them back into the patient. And doing that outside of the body limits off-target effects of the CRISPR therapy. And then after the infusion, symptoms usually disappear very quickly after the treatment. It is very expensive, however, about $2 million per patient. But it is a one-time treatment that is potentially curative, and I believe that most health insurances do cover this treatment for now. So right now, as I said, Casgevy is a great example of an ex vivo treatment where you basically take cells outside of the human, edit them, and put them back. But there's also some clinical trials going on where this is done in vivo, so directly in human, where gene editors are actually injected directly into the body. One of these examples, again, is done by CRISPR Therapeutics. They're testing CTX310 to basically remove a gene in liver cells that's responsible for producing LDL, cholesterol, and triglycerides. And so far, they have seen great success of decreasing of these cholesterol markers by about 80% with just one shot. Or Intellia Therapeutics is editing liver cells to treat ATTR, which is a rare progressive disease caused by a buildup of abnormal protein deposits in the body. We also have something called base editing. It's a next-gen method that changes a single DNA letter. So DNA is made up of these nucleotide bases. And so they basically are single letters that form a double helix. And you can usually, or not usually, but very often, some of these diseases can be caused by literally a simple letter mismatch. And so there are now these methods that are coming up that you can actually just switch that letter for the correct letter to repair the gene. And so that would be preferable, because if you don't have to cut into the DNA, it's better. It can eliminate the risk of any off-target effects. And there was a teenager in the UK with leukemia, which was treated by base-edited CAR T cells and actually went into remission in 28 days. And another company called Verve Therapeutics is using base editing to silence genes that again cause high cholesterol. So there are other things that are currently coming out that are trying to improve the CRISPR technology. For example, something called epigenetic editing. It can actually turn on or off the gene without changing the genetic code. So again, that's better than cutting into the DNA. If you don't have to do it, that's great. Companies like Omega Therapeutics are testing this for liver cancer. If you remember, I told you that currently the CRISPR system is made out of the guiding RNA, the guide RNA, and the actual enzyme, the scissors, called Cas9. But there are also other enzymes like Cas12, Cas13, and so they all have different unique precisions, and we're testing those as well. And then also AI-driven design. So again, think ChatGPT for genes. It's helping scientists to design safer, more effective edits. The bottom line is that CRISPR is an exciting new technology that can potentially provide cures for diseases we thought were incurable just a few years ago. It's single-dose treatments that can prevent certain types of cancer, heart attacks, they can treat genetic blindness, or cure rare disorders. And it can also mean more precise, personalized medicine, like what saved baby KJ, possibly could become the new normal in several years. It, of course, has disadvantages, and I didn't touch on some of the ethical issues, but one of the main disadvantages as of right now is the price. It is extremely expensive, and that might not be feasible for people that don't have insurance or people from foreign countries at this moment. But hopefully, as technology progresses, more and more people will be able to get these life-saving treatments. Thanks for watching, and I hope you liked the video. Give it a thumbs up, and please subscribe for more biotech deep dives.